Hi everyone, and welcome to Chapter 11, Part 1b on the maturation and activation of lymphocytes. Yes, I know you have heard about lymphocytes already. We heard about it in last video. We also heard about it in Chapter 8, but I'm telling you, this is way more detailed. Now, before we go into lymphocytes, we need to talk about what happens to our body every single day. Every single day, we are exposed to so many different types of pathogens. You don't know it, but our body is constantly fighting a war here. Now, but there are so many million types of pathogens. There's so many out there. And if you were a smart, country, you would have many special task forces to combat the many enemies you have, right? So the same idea here, your body will have many types of B cells and T cells specific for different antigens. If there are millions of pathogens out there, it will have million types of billion B cells and T cells. Now, each type of lymphocytes would respond to only one type of antigen only. This is why we say that lymphocytes are involved in specific response. It is this specific. For example, if you have one type of B cell, it will only produce one type of antibody receptor. So like this Y shape, just one type, not just one. There are many Ys around the cell, but you get it. There's only one type of antibody receptor, and this one type of antibody receptor would respond to one type of antigen only. Don't worry about the term antibody receptor. I'm going to get to that later on. But the point is, if we have million types of B and T cells which are specific, this means that our body can respond specifically to almost any type of pathogen out there. We are ready for the war. Now, that is about the millions of different types of B and T lymphocytes. But the reality is, they don't just sit there and do their job and that's pretty much it. There's actually a growing process, a maturation process, an activation process for lymphocytes. Only the mature lymphocytes can actually circulate in lymph and carry out the immune responses. Okay, so what do we mean by maturation? This is what we mean, okay? They are, once upon a time, there is stem cells in the bone marrow. All your T cells and B cells are produced before birth. But these B cells and T cells are naive. They are not mature yet. They are naive B cells. B cells would mature in the bone marrow. Made in the bone marrow, mature in the bone marrow, become mature B cells. When the pathogen that it is specific to attacks the body, then it will get activated to form plasma cells, which are still B cells, but a specific type of B cell. These plasma cells can produce antibodies. And also, mature B cells will differentiate to become B memory cells, which does a different function. How about T cells? T cells, also the same idea. They are made in the bone marrow from the stem cell, forms naive T cells, and it undergoes a maturation process in the thymus to form mature T cells. Then it can circulate in the blood and lymph. But it doesn't really differentiate and activate until the pathogen that is specific for attacks the cell. Then it's activated, and it differentiates into T helper cells. T helper cells later on can differentiate to T helper memory cells. Mature T cells also can differentiate into T killer cells, or what we call cytotoxic T cells, which then also differentiates into T killer memory cells. So this chart here is very useful, and I recommend that you sort of remember these types and remember this chart so that the future processes that I'm going to talk about around now, it's going to be easier for you. Okay. We are going to be talking about B lymphocytes first, and then the T lymphocytes. So all the B cells would have a 1 at the left hand corner here, and all the T cells related would have a 2 in blue at the top left corner here. So let's talk about it in more detail. Let's go. Maturation of B lymphocytes. Now again, all cells are formed in bone marrow before birth. It was formed in your bone marrow before you were born. What happens there? There are millions of types, right? So you expect that genes in B cells code for different type of antibodies for different type of B cells. Now, each B cell will have a specific antibody which acts as a glycoprotein receptor. Antibodies are glycoproteins, which means it has a protein, it also has a carbohydrate attached to it. And this is on the cell surface membrane of B cells. And the specific antibody receptor will bind to the antigen that is complementary in shape. This gives the specific B cells its specificity. Yeah. So one type of B cell has one type of antibody receptor, can only respond and be activated by one type of antigen. After that, B lymphocytes can divide and mature in the bone marrow, and these mature B lymphocytes can circulate the blood and concentrate in liver, spleen, and the lymph nodes. Now, let's talk about antibodies. When we say antibodies, what do we mean? You now know that it's a glycoprotein, okay? And here's more information. It is also globular. The carbohydrate part is usually not shown in diagrams. You only see the protein parts here. Just so you know, another name for antibodies is immunoglobulins, and sometimes it can be summarized into the abbreviation IG. So immunoglobulins or IG. Anyways, what is it made of? What kind of level of structure it has? It is made out of 4 polypeptide chains. And this is just a representative diagram, but generally all antibodies are represented by this Y shape here. It's 4 polypeptide chains, 2 heavy chains which are in blue here, and 2 light chains, that is green. Now because it has 4 polypeptide chains, this obviously is a protein with a quaternary structure. Don't forget it has a glyco part which is not shown in the diagram as well. How is it held together? It's held together by disulfide bonds which give it stability. There are 3 regions on the antibodies. Number 1 is a variable region. Now, variable region is these parts here in yellow. There is a light chain variable region here, and a heavy chain variable region here and here. So they are formed by both heavy and light chains. This variable region is important to provide 2 identical antigen binding sites. So this is one binding site, this is another antigen binding site, and that means each antibody has 2 binding sites lah. It is specific for binding antigen as this particular part is complementary in shape to the antigen, and the shape here is determined by the specific sequence of amino acid. You can see here that it's a little bit jagged, and this is to show you that hey, this one has a specific site for specific antigens. The R groups at the antigen binding site would form hydrogen bonds and ionic bonds with the specific antigen that it binds to. Now, again, it's specific, which means that the sequence of amino acid at the variable region is different for each type of antibody. Each type of antibody would bind to different antigens. Again, one type of B cell, one type of antibody receptor, okay, one type of antigen. So different B cells will have antibodies with a slightly different primary structure, slightly different specific sequence of amino acid at this variable region. So this one differs from different B cells, different type of B cells. Now, the constant region, however, here in blue, the primary structure of the protein or the sequence of amino acid is the same. So blue regions, again, sequence of amino acid is the same for all the antibodies, all the different types. But a variable region for different types would have a different sort of sequence of amino acids. So speaking of constant region is formed by heavy and light chains, just like the variable region. And what does it do? When it circulates in blood, okay, when antibodies are circulating in blood, these antibodies can actually bind to receptors on phagocytes. So you can see this picture right here. You can see that here the antibodies, these Y-shaped things, are bound to this antigen or pathogen here, and then the constant region of the antibody is actually binding to a receptor on a phagocyte, and this helps the phagocytes engulf the pathogen. Now, but when the antibody acts as a B cell receptor, so something on the B cell here, an antibody receptor, this constant region is the region that attaches to the cell surface membrane of the B cell. So there are two functions here, depending on the question, depending on the context. One is when the antibody is freely circulating, the other is when it's acting as a receptor. Now, another function of the constant region is to really give the antibody different classes. No, it's not the same as your M1, M2, S1, S2, S3 sort of thing. No, different class as in types. So you can see here IgM, IgG, okay, these are immunoglobulin M, okay, short for that. And they have sort of different constant regions, okay. You don't need to remember this, I'm just showing you what different shapes antibodies can take. Now there is a third part here for antibodies. There's the variable region, there's the constant region, and then there is the hinge region. The hinge region is pretty easy. It is held by disulfide bridges. It is this area here. And what it does is just gives some flexibility when binding to antigens. That's about it. Now, let's talk about the action of the antibodies. We saw a little bit of the action just now. This is the same picture here. But let's do sort of a range of different things. Now, these parts here are important, but I feel like the textbook did a very good job at making this diagram, so I just copy pasted it and added some titles. Okay, so actions of antibodies. How do antibodies work? These are antibodies that are freely circulating. Okay, now, antibodies can prevent entry into cell. Okay, it can bind to the virus or the toxin and prevent it from getting to the cell. It can attach to flagella of bacteria and prevent it from moving around. So if it's less active, it is easier for the phagocytes to engulf. Number 3, it can bind, agglutinate, it can clump together those bacteria or pathogens, so that it's also easier for it to engulf and restricts the movement. So this is called agglutination. Number 4, it can cause pathogens to lyse. So it actually can poke a little hole, and when water moves in, the pathogen can lyse. Number 5, they do something called opsonization. Opsonization is the process that I described you just now. The antibody would bind to the receptor of the phagocyte membrane, okay, and facilitate the engulfment of a pathogen. Makes it easier for the macrophage to find it and to ingest the pathogen. And number 6 is to neutralize toxins. So freely floating toxins can affect our cells and enter our cells. So by binding it, it makes the toxins not toxic. So some people say that antibodies also act as antitoxins. So that was antibodies and sort of the maturation of B lymphocytes. If it's too long didn't hear or didn't read, B lymphocytes, they produce in the bone marrow, they mature in the bone marrow. Each of them have an antibody receptor of a different shape on their cell membrane. Antibody structure and action you need to know. But when exactly is antibody secreted? When exactly is it acting as a receptor? What? When? How? Okay, we'll get there here. So let's talk about the action of B lymphocytes. This is after maturation. After maturation, let's say there's a pathogen that invades the body. What will happen first is that the antigen presenting cell will form. So this could be a phagocyte, maybe a macrophage, engulfs the pathogen and presents the antigen to the B lymphocytes. Okay. Only the specific B lymphocytes has receptors with the complementary shape to that particular antigen will be activated. Because there are many types of B lymphocytes, right? Only this specific special task force will be activated. So this is selected. So that's why it's called clonal selection. This activated B cell then divides by mitosis, and this is called clonal expansion. So you will result with many of the same type of B cells. So one B cell that is activated become many activated B cells. Now, activated B cells then can develop or differentiate into plasma cells and memory cells. Let's talk about plasma cells. Plasma cells are short-lived, they are only a few weeks, but their main function is to produce and secrete antibodies rapidly. They produce a lot, and they do this by exocytosis. Okay, again, antibodies are globular glycoproteins, which means they will have to undergo modification in the Golgi and then secrete by exocytosis, something you have learned in chapter 1. Secrete to where? Secrete to blood plasma, lymph, lungs, and stomach linings, and it's going to circulate in the blood. Now, because this requires a lot of energy and a lot of processing, you expect plasma cells to have a lot of ER and Golgi as seen here. That's plasma cells. Now, again, the activated B cells also differentiated into memory cells. So what do memory cells do? Memory cells are long-lived, they remain in circulation in the blood and lymph, and it actually provides long-term immunity. That's why it's called memory cells. And what they do is they last for many years in lifetime, and they enable a faster response during the second invasion of the same antigen because they are memory cells circulating already. During the second invasion of the same pathogen, it can divide rapidly, okay, or clonal expansion. It can form more plasma cells, which forms more antibodies, and this speeds up the response. And the infection is actually destroyed before symptoms develop. This is where we call where we say that the host already has immunity, or the host is immune towards a certain thing. Maybe because the host has been infected with the same thing before or similar thing and has memory cells in the body. Now this is a graph of what memory cells does. This is what happens during the primary response. You can see that this is a graph of concentration of antibody against time. The primary response is a slower response here. Okay, quite a less steep graph compared to this. This is because only a few B cells specific to antigen is present, and as a result, the individual does become ill and takes some time before the individual becomes healthy again. However, memory cells are produced here, and during the second exposure to antigen, you can see that it's a steeper curve, faster response, and actually a bigger response. You can see that concentration antibody is much higher here. And this is because many memory cells are circulating only, so it detects it faster. There's a higher chance of encountering those pathogens quickly. More plasma cells are formed compared to the primary response, and therefore even more antibodies are produced in the response. As a result, the individual do not develop any symptoms. The pathogen will be killed and dealt with even before symptoms develop. So this is a summary of action by B lymphocytes. First of all, they mature. Second of all, when the pathogen invades, there is binding, okay, with the specific antigen on APC. This is called clonal selection. And then there's division by mitosis. This is called clonal expansion. And then there is differentiation into plasma cells, which produce and secretes antibodies here into the bloodstream. And also, there will be the formation of memory cells, which enable the faster response during the second invasion of the same antigen. And that's it. That is the summary of action by lymphocyte, B lymphocyte.